Good day, dear students. In current lecture, we'll discuss infections which are known as zoonotic or zoogenic infections. We will discuss most important of them. So first of all, of course, we need to say which infections or which types of infections are possible in general. According to international classification, we have three main types: sapranosis, anthrapanosis, and zoonosis. I gave you definitions of CDC Center of Disease Control and Prevention. They are most simple and the most easy to understand. But in general, they all are equal. So sapranotic infections or sapranosis, they are infections which are observed in some abiotic environment and can be accidentally transmitted to human. It can be any environment in surrounding us, nature, some decaying plants, water, or some other. And usually they are rare in community, or they can be observed in people with impaired immunity. Most popular type of that infection is mycosis. It can be visceral or skin mycosis, which is very rarely observed in immunocompetent people, but in people with HIV infection or other conditions. Those types of infection are very common. Anthrapanosis or anthrapanotic infections. It is a disease which is transmitted from human to human and can be observed only in human population. We have very limited number of those infections such as diphtheria, gonorrhea, or rubella, but they are very dangerous and they are very important because can be easily transmitted from person to person contact. And we have zoonotic infections or zoonosis. In common, it is type of disease which are naturally observed in animal population and can be accidentally transmitted to human. And those infections are very dangerous because common community of people have no immunity against those diseases. They are not natural for human. And some authors, some scientists say that arthropod-borne diseases, it is a unique type of disease, but actually they can be transmitted from human to human through arthropods or through from animal to human. So it's not actually a separate type, but anyway, it is unique type of infection. It is type of disease which is transmitted through blood-sucking insects. And we have again such famous example as malaria or typhus, epidemic typhus, which is arthropod-borne type of disease. So if we will discuss zoonotic infections or zoonotic diseases, they have again two main groups. Zoonosis themselves formerly were called as arthropozoonosis because actually they are transmitted from animal to human if we will talk about human disease. And we have zooanthropanosis. It is type of disease which is transmitted from human to animals. Those are very rare and we will not talk about them, but anyway, why those diseases are important and sometimes even dangerous? Because main number of them have very high mortality rate. A lot of people will die because again the disease is not natural for human and we have no natural protection against those diseases. Some of diseases can be transmitted from patient to hospital personnel to doctors to you. And of course we have few but still extremely important examples when zoonotic diseases caused extensive outbreaks, epidemics, even pandemics with very high mortality rate. We will discuss them today. And actually in current lecture we should explain such important and popular topics as extremely dangerous or highly contagious disease, or more correct, and I like the definition most of all, high consequence infectious disease. It is use of UK government, but still very good explanation and criteria. So what is it? Extremely dangerous disease. Of course, it's not a cancer as a lot of you say, and it's not some common type of disease which is not actually an infection. Of course, when we're talking about high consequence infectious disease, that disease should be an infection. And it can be any infection which is mentioned following criteria. It have acute onset and acute duration, typically associated with very high fatality rate. And of course, if it is high consequence infectious disease. It should not have effective ways of prevention and treatment of that disease in the case of massive spread. Often difficult to recognize, we have no specific methods of fast diagnosis and fast recognition of that disease. It should be able to spread with very high speed in community, even within the healthcare settings when doctors who should help will be affected one of the first. And of course, that type of disease should lead to system response, require system response to prevent further spread of the disease. Why are we talking about high consequence infectious diseases? Of course, because if that disease infection matches those criteria, that infection can be used as a biological weapon or biological warfare. And again, we have a lot of different definitions and criteria for biological warfare. In current slide, I show you some criteria of WHO, and you should understand that biological warfare is some type of agent. It can be a bacteria, virus, or toxin produced by that agent, or some other substrate originated or obtained from the microorganism, which have ability to cause a disease with little outcome in human or even animals. And of course, we're talking about that thing because very high possibility that some bacteria can be used as a weapon or some weapon of massive destruction by terrorism, or it was a very popular topic to discuss when we had the Cold War between USA and USSR. And of course, a lot of countries have biological laboratories with number of pathogens can be used as a bioterrorist attack or as a weapon of mass destruction of population. So, of course, it is not really high chance that some bacteria or virus will be used against people, but still it is a chance. And of course, you as a future doctors will be the first line of contact with people who suffered from that type of weapon, and you will need to recognize and to provide the medical help. And of course, you will be in very high risk to be infected with those agents. And if you will take a look on criteria for biological weapon, and you will compare them with the properties of zoonotic infections, and you will understand that actually definitions are matching. Because in the case of zoonotic infection, they always occur in human as acute infection. They are associated with high case fatality rate because people have no immunity against that disease in natural way. And of course, we don't have effective prophylaxis and treatment measures for massive spread of the disease, because those diseases naturally are very rare in community. And of course, no one will think about some zoonotic infection in human in routine way, and that disease will be difficult to recognize, but it can very easily spread in community and affect even doctors as well. So we have a lot of examples in human history when bacteria really were used as a biological weapon. First dated use was about 1,000 years ago, and again Italian warriors used human bodies, dead human bodies, to poison water of enemies. And second very important and famous use by Mongols, they used plague victims. They throw them with the help of catapult over the city walls, and bacteria were released after contact with after contact of dead bodies with healthy citizens, and a lot of people died because of plague. And multiple examples of use of different bacteria and viruses by different countries and communities against other people. Of course, nowadays we have several conventions which prohibits use of any biological weapon for massive destruction of people, but still it is an option in the case of some bioterrorism or even war conflict between different countries. So first disease and first bacteria we'll discuss in current lecture is plague, which is caused by Yersinia pestis. We already talked about that bacteria when we discussed food poisoning and food infections. We discussed Yersinia. You should remember that it is Enterobacteria, and genus consists of multiple species, but only one, Yersinia pestis, can cause plague. Bacteria mention all requirements for Enterobacteria. It is gram-negative, non-motile, and non-sporin rod with facultative anaerobic respiration. It can have capsule, or very often it is called as a slim layer because it is very thin. And in slides it can be pleomorphic, means it can have different shapes. And again in the right slide it is stained with the methylene blue or Giemsa stain when bacteria have unique shape of safety pink, or it's bipolar staining when center is transparent and both poles are stained in blue. So bacteria is again naturally found in animals, and natural reservoir is rodent and fleas. And body temperature of fleas and rodents are lower than in human, and optimum temperature for cultivation of Yersinia is 27 degrees Celsius, and of course it has very slow growth because of unique requirements. We have several registered cases, recent cases of plague even in peaceful time, and we have natural reservoirs of those bacteria. We have very intense, severe spread of pneumonic plague. It's still continuing in Madagascar, and over 200 deaths are registered. Very difficult and not really possible to stop that epidemic. And of course, for our benefit, it is an island and disease not spreading to other countries, other cities. It is limited to Madagascar only, but again it is extremely dangerous problem still to different World Health Organizations. So for cultivation of bacteria, again Enterobacteria, not very difficult cultural requirements. McConkey agar is preferred culture media for cultivation of any Yersinia, and colonies will be lactose negative, very small, colorless. If you will use blood agar for cultivation, you should have white colonies with black or dark brown zone around colonies. Agar will be changed in color because of hemian absorption. As any Enterobacteria, it should be differentiated with the help of Kligler iron agar, and it have lactose negative and glucose positive result, no gas, no H2S, but production of acid, which will change color of lowest part, tube part of agar, into yellow. Factors of pathogenicity are not very difficult. We have few extremely important. It is F1 antigen or fraction 1 antigen. It is an enveloped protein which have ability to inhibit phagocytosis, and it is produced by bacteria only in 37 degrees Celsius. So if we will take environmental sample or sample from animal, from rat or from flea, that antigen will not be identified because temperature is not sufficient. But if it is sample obtained from human, then that antigen is produced and easy to identify. Two additional antigens, V and W antigen, both have similar properties. V antigen is more important and used in diagnosis and prevention of disease. Both are responsible for inhibition of phagocytic activity of our immune cells and resistance against intracellular killing by our phagocytes. Again, we have mutant strains. Strains are used for production of vaccines. Bacteria can produce important bacteriocin, which is called pesticin because of unique name of bacteria. and it is responsible for inhibition of other species of Yersinia or even E. coli. It can be important because after ingestion of Yersinia pestis, bacteria will not be inactivated by our normal flora. It have unique weapon against our normal flora E. coli. Cardiotoxin is described in that bacteria have confirmed effect in animals, and it is cardiotoxin. It can affect and block activity of the heart, but activity in human is not confirmed. Maybe it have some effect, but it's not really important because of other complications in the case of plague. And one unique enzyme which is known as coagulase or block enzyme, it is essential for transmission of disease from flea to rats and from flea to human. Again, it is produced at 27-28 degrees Celsius. It is normal temperature of rat fleas. If you will take again sample from human, that enzyme will not be identified, but in animals easy to identify. According to international classification, we have three main forms, clinical forms of plague, but actually those forms, they should be as well recognized as different stages of plague in common. So we have bubonic plague, septicemic, and pneumonic plague. Most popular and most common type of disease is bubonic plague. Bubo is affected lymph node, black and tense and painful. So pathogenesis will be described in the next slide. A little later we will discuss, but in common, the main symptom of bubonic plague is inflamed and affected regional lymph node. Regional means closest to the site of the bite, and that lymph node is called bubo, and type of disease is bubonic. Very often lymph node can turn into separative sore with ulcerated open area, and the main idea that bubonic plague itself can be transmitted only by bites of fleas. It cannot be transmitted from human to human, and if some subject is affected by bubonic plague only, then that subject is not dangerous for hospital personnel, for doctors, if proper requirements will be followed, such as use of gloves and other. Septicemic plague, usually it is the second stage of bubonic plague. Bacteria will invade into the bloodstream and spread, but very often that disease can be observed as a separate type of disease when some damages on the skin, open wounds, or trauma during surgery or medical help with common needles or some instrument. Then bacteria can invade directly into bloodstream, and septicemic plague will be observed without previous bubonic plague. And the last stage of disease or third type of infection is pneumonic plague, when lungs are affected, and it is the most virulent, the most deadly form of the disease. Again, typically it is the last stage of bubonic plague. With the bloodstream, bacteria will invade into the lungs, and you will have pneumonia in those people caused by plague. But people with even secondary pneumonic plague who already suffered from bubonic, septicemic, and now pneumonic plague, they are highly contagious for environmental people. They can produce aerosol, and it can spread over the air and infect everyone around. And disease is highly contagious in the case of pneumonic plague. So that type of disease is almost always fatal, and very few cases of cures were described in history, but nowadays mortality rate is higher than 99%. So first of all, we will of course discuss bubonic plague, which is a very common and the most common type of the disease. Again, disease transmitted by bites of insects, and only infected flea or rat with the fleas can be the source of infection. Bubonic plague cannot be transmitted from human to human. That type of disease is only for pneumonic one. So way of transmission of bubonic plague as a vector-borne or through blood-sucking insects. In current age, it is a rat flea. So naturally, disease is transmitted by bite. Initially, rats are natural reservoir of the disease, and disease transmitted in rat population with the help of rat flea. Bacteria will invade into flea and multiply in the gut. In the gut of the flea, bacteria will produce that coagulase block enzyme, which will cause a blockage of digestive tract of the insect, and that blockage will prevent sucking of the blood. And when flea will bite another rat or another victim, it cannot ingest the blood, and blood will be mixed with the bacteria, multiply in the gut, and will be regurgitated back into the bite wound, and new victim will be infected with that bacteria. So flea of course will be very hungry, and it can bite a lot of different victims in very short time period. And usually disease is observed in very poor hygienic conditions when rats are very close to human and sanitary condition is poor, and of course hungry flea will not choose whom to bite, rat or animal or even human, and disease can be accidentally delivered to human. And of course in the case of epidemic, disease can be transmitted from human to human, but again only by bite of insect, not by direct contact with that person. So after bacteria is ingested by flea and flea bited healthy man, bacteria will invade into the wound. In wounded tissues, in skin or even underlying tissues, of course, very first cell to recognize the pathogen will be phagocyte, and phagocytes will engulf bacteria. But Yersinia pestis, due to again VMW antigen, can resist phagocytosis, will survive and multiply initially in our macrophages. After that, bacteria will be delivered to regional lymph nodes, and here it will continue multiplication but in very high rate. It will lead to formation of intense hemorrhagic inflammation with enlargement of lymph node, and later stages it will undergo necrosis. It will become blackened, and again that type of blackened inflamed lymph node is called as a bubo. Of course it will manifest with very severe edema in affected lymph node, fever, pain, and usually it is a leg when men are very high and flea cannot jump very far. It will bite into the leg, and inguinal lymph node is affected most of all. Of course if man was lying or sleeping on the ground, it can be neck, arms or other body parts, and regional lymph node is not always inguinal, but it is most frequent. It will lead to affected motility. People cannot step on the leg, they cannot walk faster, they will usually looking for medical help because of severe pain in inguinal region. So after lymph node is necrotized and destroyed, natural protection is broken, bacteria will spread into the bloodstream and cause septicemia as the separate stage of the disease. Very severe state when massive release of bacteria will lead to massive release of endotoxin or LPS, and in Yersinia the toxin is less active than in other enterobacteria, but still in intense amount it will lead to common symptoms such as phobia, loss of powers, hypertension, and because again different type of hemolytic enzymes, it will lead to different types of anemia or even disseminated intravascular coagulation syndrome, and discoloration of extremities changed with blackened of the extremities. In severe stages it will lead to altered mental status. People will not understand where are they, what's happening, and mental ability will be lost. So as the fatal in the last stage, multiple necrotic lesions will be observed in all organs including skin, and that unique black ulcers of skin gave unique name for the disease, and history it is known as the Black Death. So in final stages, most susceptible organs affected by bacteria will be kidneys. It will lead to acute renal failure. Sometimes it can lead to heart failure because of multiple organs affected, and blood circulation will be affected overall. Again, it is possible to have pneumonia in those people as the type of complication after bubonic form or primary pneumonic form if patient was in direct contact with other people, healthy one. So in general, bubonic form of disease have a mortality rate about 50%, so every second person will die. But in the case of pneumonic and septicemic plague, mortality rate is very close to 100%. Again, very few cases described with the curage. So in the case of proper treatment, and of course in the case of bacterial disease, it should be antibacterial treatment such as streptomycin or tetracycline. Mortality rate can be decreased till 15% or even lower. But again, in the case of massive spread of the disease, when a lot of people are affected, of course, hospitals will not have sufficient supplies of those antibiotics. They are not very popular, and number of them in hospital are limited, and treatment of that disease will be very, very difficult. Few pictures of bubonic plague in the beginning and in the late stage. In the beginning it is just enlargement, and in the late stage it is blackening with formation of ulcers, and you can see even blood vessels affected with bacteria, and of course state of people is very severe. So if you need to diagnose disease, your tactic is dependent on the stage of the disease or type of the disease. In the case of bubonic form, of course, first of all, you need to take some material from lymph node. It should be aspirate or biopsy of lymph node. If you suspect that there is already bacteremia, you need to take blood. And in the third stage, when bacteria is delivered to multiple organs, of course, you can take any material, cerebrospinal fluid, sputum, or some other. Of course, disease is extremely dangerous to recognize as soon as possible. And we have several dipstick tests with the use of monoclonal antibodies, which are targeted against F1 antigen, and tests can be performed near the bed of the patient. You don't need any specific equipment. It is type of immunochromatography test, paper strip with few reagents. You need to take sample, add a drop on the test, mix with reagents, and in 15 minutes you will have a result, positive or negative. Of course, monoclonal technique is very precise and result is for sure. So again, disease is registered in very, very limited cases, very limited number of cases, but still it is registered daily worldwide. And one of the chief reservoir area is India. So in your country that disease is important even in peaceful time. We don't have any specific prevention, but again, disease is vector-borne and transmitted by fleas. So common measures to control animals, to perform deratization and control of insects is very sufficient measure to prevent massive spreads of the disease. Of course, we have some vaccines we will discuss later, but routinely they are not used in schedule of vaccination, and they indicated only for travelers in epidemic areas or people who are working with bacteria, or for doctors who are working in endemic areas. Those vaccines are applied. So again, we had massive pandemic of plague in mid of 13th century, and according to different data it is difficult to evaluate now because very few papers are safe and available nowadays. But according to different data, about a quarter of world's population died because of that plague disease. Again, historical name is a Black Death. We have several pictures and wax sculpture with description of the disease. And of course, you can see that half the person is healthy, not affected, and half of the person is in the last stage when the whole body is affected. And people were all covered with black ulcers, small to large, different size. They were bleeding, released some necrotic discharge. People were were coughed with the blood, and a lot of people died in really painful manner because of the disease. And disease is very dangerous and severe even nowadays. And of course, you all saw that Plague Doctor. It is one of the most famous presentations of something dangerous and something scareful. It was first used in Rome by Romanian doctors. A specific type of oil where put it in that nose, and glass protect eyes, and doctors thought that the specific aroma oil will save them against bacteria. Of course, not an oil did safety thing. It is type of mask which have mechanical protection of air, and of course it was helpful, but not always. A lot of doctors still died because of plague. There were infections from people, but that picture is still used in multiple movies, animations, and other. So, if we'll talk about immunobiological preparations which are used to diagnose or prevent plague, of course, again, first of all, it is type of immunoglobulin against F1 antigen, which is used for rapid detection of bacteria in clinical sample, any type of clinical sample, or in culture medium if essential. It is simple immunofluorescence. You need to apply immunoglobulins labeled with fluorescent dye with your clinical sample, and if that sample contains Yersinia pestis, antibodies will cover bacteria, and in UV microscope you will see intense shining, glowing as the shape of bacteria, very small cocobacilli or rod shape. We have specific human antibodies, monoclonal antibodies against V and F1 antigens, which are used for treatment or passive prophylaxis after contact with confirmed case of plague, pneumonic or other. Again, technology is very promising and we have perfect results, but still only in mice experiments. And even if it is human antibodies, it is difficult to study them because number of people infected with plague is very limited. And of course, no one will allow to infect human with the plague with the purpose just to check activity of those antibodies. So data in human is absent, but again in animals results of those antibodies are very promising. And again, in the case of some accident and if plague will appear again, that type of therapy of course will be used and the results will be evaluated. We have one type of vaccine which is approved and used worldwide. It is type of plague vaccine, inactivated vaccine. It is inactivated with formaldehyde and preserved in phenol. So it is one of the very first vaccine. It was used since the end of 19th century, so more than 100 years it is used for prevention of the disease, but nowadays that vaccine is prohibited for using human by several reasons. But again, common indications for plague vaccination is intense work with bacteria in laboratory conditions. It is person who work with animals in epidemic or endemic zones, some control of rodents or cleaning of some environmental areas. And of course, if some people, some veterinary doctors or other people who have direct contact with rodents or rabies, they have high risk to be accidentally bited by flea. They should be vaccinated as well. So three main indications for vaccination are known till nowadays. Schedule of vaccine is kind of difficult. It differs for different age groups, and people, adults and older than 10 years of age should receive 3 doses of primary vaccination, different volume in 0 day means today, in 4 weeks later, and in 6 months later. Again, if it is some single indication for vaccination, you need to just a trip or you have short time period for job activity, then 3 doses are final. But if risk is still present, further booster vaccination should be performed every 1 or 2 years depending on degree of exposure. Again, for kids 10 years of age and before 10, we have the same time interval, but doses are different. Lower the age, lower the dose. Again, booster vaccination, if required, should be performed as it's shown in the table. Again, inactivated vaccine is the only type of vaccine which is still used in some countries, and it is the only approved by CDC and the international organizations. But we have number of vaccines, and actually large number of vaccines in research and development stage. One of the most popular type it is subunit vaccine, mainly obtained by genetic engineering, when genes of Yersinia pestis are introduced in some other bacteria, especially E. coli. Again, some companies trying to design vaccines for peroral use or subcutaneous. They are more safe and have less number of side effects. And some companies are trying to design combined vaccines for prevention of plague and anthrax together, but again data is limited and only some studies on animals or even in vitro studies are available. We have number of attenuated strains, and mainly we have attenuated vaccines, and mainly we have two types. It is a mutated strain of Yersinia pestis itself, or we have some recombinant vaccines with Yersinia pseudotuberculosis. Again, we will talk when we talked about the bacteria in previous lectures. We said that Yersinia pestis and Yersinia pseudotuberculosis are very similar, and genetic combination of bacteria is 95 and even higher percent identical genes, and even some virulence plasmid, one of two, is identical in two bacteria. So some companies trying to use Yersinia pseudotuberculosis as a vaccine strain to prevent plague. And we have modern and most promising type of vaccine, which is called vectored vaccines, when some not dangerous bacteria is used to deliver genes of Yersinia pestis. Again, a lot of different studies, a lot of data is available, and such bacteria as a Salmonella or even Lactobacillus, some companies trying to use viruses, tobacco mosaic virus, to deliver genes. And one of the most popular type of vaccine is intranasal vaccine, when some aerosol is inhaled in nasal cavity and provide very good immunity. Again, no injection is needed. It's not required to have some medical nurse or other personnel. And one of the most promising and the latest directions in vaccine and vaccination in general, in the case of Yersinia pestis included, it is OMV vaccine or outer membrane vesicle vaccine, when specific organelle, not even organelle, it is specific structure of gram-negative bacteria, vesicle, which have very small nanocytes, is used to deliver genes of pathogenic bacteria into human body. 2 years ago, in 2018, in workshop of WHO, a group of research scientists, they showed vaccine which is based on vesicles of Bacteroides containing V antigen of Yersinia pestis. And again, results are available only in animals, and results are very promising, but still no type of vaccine got some governmental permission to be studied and tested on humans.